Assalamu alaikum. My name is Atiya Banfal Jilani and my roll number is EBP 1901016. And my assignment topic is differential leukocyte count, which is known as DLC count. So, what is a blood differential count? The blood differential test can be detect abnormal or immature cells. It can also diagnose an infection, inflammation, leukemia, and immune system disorder. It measures the percentage of each type of WBC present in the blood. A blood differential test is often part of a complete blood count CBC. A CBC is used to measure the following components of the blood: white blood cells, which helps to stop infection; red blood cells, which carry oxygen; platelets, which help clot the blood; hemoglobin, the protein in red blood cells which carry oxygen; hematocrit, is the ratio of red blood cells to plasma in your blood. A blood differential test is also necessary if your CBC are within the normal range. And then we have type of WBCs. WBCs or leukocytes are classified into two main groups: granulocytes and agranulocytes. The granulocytes, which include neutrophils, eosinophils, and basophils, have granules in their cell cytoplasm. They also have a multi-lobed nucleus. As a result, they are also called polymorphonuclear leukocytes or polys. The nuclei of neutrophils also appear to be segmented, so they may also called segmented neutrophils or segs. Then we have the non-granulocytes. White blood cells, lymphocytes, and monocytes do not have lobules and have no non-lobular nuclei. They are sometimes referred to as mononuclear leukocytes. Then we have a chart of the type, type, appearance, micrograph, and illustration. And then main targets, granules, lifetime. For example, we have first type, which is neutrophil. So it is present in adults approx 62%. Its diameter is 10 to 12. Its main target is bacteria and fungi. Its nucleus is multi-lobed. Its granules are fine, faintly pink. Its lifetime is 6 hours to few days. Then we have another type, eosinophil, which is present about 2.3% in adults. Its diameter is 10 to 12. Its main target is larger parasites and modulate allergic inflammatory responses. Its nucleus is bilobed. Its granules are full of pink, orange, and its lifetime is 8 to 12 days. Similarly, we have another type, which is basophil, and it is present approx 0.4% in adults. Its diameter is 12 to 15. Its main target is to release histamine from inflammatory responses. Its nucleus is bilobed or trilobed, and its granules are large blue color. Its lifetime is few hours or few days. Similarly, we have lymphocyte and monocyte, which have the same criteria. And then we are going to another slide, which is requirements. So Leishman's stain, it consists of a mixture of eosin, that is an acidic stain, a methylene blue, that is a basic stain, in alcohol and is usually diluted and buffered before use. It stains the different component of blood in a range of shades between red and blue. Glass slide and glass dropper, distilled water, sterile lancet, cotton and gauze swabs, 70% alcohol, microscope. Then we are going to its procedure. First of all, we have preparing the blood smear. Sterilize the fingertip of the subject with the cotton swab dipped in 70% alcohol and is dried. Take a blood Take a bold prick on the fingertip to have free flow of blood. Then collect drop of blood on the end side of the glass slide. Then spread the blood drop with another glass slide by placing it an angle of 45 degree and move sidewise. Hold the spreader firmly and move it on the previous slide to the other end in a straight line with same force and pressure. Allow the glass slide to dry after formation of the smear. Then we have second step, which is staining the slide. Keep the smeared glass slide on the flat surface with the smeared surface facing upward. Pour drops of litmus stain on the glass slide to cover the smear or film. Keep it undisturbed for 2 to 5 minutes. Pour drops of distilled water on the slide and leave it for 10 minutes. Then remove the dye and water. Remove the extra stain by keeping the slide under running water. Then keep the slide aside for some time to dry. And then we have third step, which is observation of glass slide and counting of cells. Keep the prepared glass slide is under low power of compound microscope and choose a good quality slide. Then identify different types of WBCs under medium power. Draw a table with 10 boxes or horizontal and vertical axes on observation notebook. Fix the slide on the platform and choose the area towards the corner. Note the different types of WBCs found on the table in an abbreviated. Move downward and in chain-like manner till 100 cells are observed. After counting 100 cells, prepare the report. Here is the observation. We can see the WBCs are present here. Basophils, neutrophils, lymphocytes, monocytes, eosinophils, all are present. Thank you. This is my part, and the other part will be inshallah ta'ala described by my partner. Thank you so much.